Welcome, everyone. We have today have experienced so far, I think, a really wonderful Young Investigators meeting. It was great listening to all the presentations and the discussions between the young investigators, the parents, and I'm hopeful that the grant mechanism panel was very helpful for you. I am Connie Smith-Hicks. We have with us today Drs. Holder and Schläger, who will be joining me in talking about our experiences on our path thus far. We'll each speak for probably about 5-10 minutes, and then we'll have an informal sort of a question and answer period. We thought that since we were such a small group, we would have this more of an informal conversation. And so maybe we can have the trainees sort of tell us a little bit about themselves. And Amina, how about we start with you?
Sure. Hi, I'm Amina Smith-Fine. As you can see from my background, actually I'm at Kennedy Krieger. I just finished up my training in neurodevelopmental disabilities there. As the 4-year child neurology and DD between Hopkins and Kennedy. And working, so as a fellow, started working in the lab with Ali Fatimi in the Moser Center for Leukodystrophies. I'm very interested in leukodystrophies and in neurogenetics research and clinic. And so as of finishing training in June, in July, I started a faculty position as an assistant professor. And most of my time currently is spent on with clinical research, but I have a couple clinics as well in the Center for Development and Learning and in neurogenetics and leukodystrophies, which is mostly ALD and AMN. I'm currently on, later into maternity leave. So that's what I've been up to most recently, but that's basically it.
Congratulations.
Thank you.
I hope you're enjoying your time with her.
Thank you. Yes, absolutely. She's sleeping in the background, so my mom is here for help, but it's possible that I'll have to end a little early.
That's okay. We understand. And Nathan and Kristen, do you want to tell us a little bit about yourselves?
Kristen, go ahead. I'll follow you. Or I'll just go. This is Nathan. Full disclosure, I'm actually on the board at Bridge the Gap. So I'm more or less just a little set of ears for Monica and Chuck, but I am a physical therapist by background who's been in neuroscience research for, I don't know, nearly 20 years now, obviously more at the human level. I have a company named Rehab Metrics, who is in the rare and orphan disease space. We do a lot of the performance and functional type assessments for gait assessments, movement analysis, that kind of stuff, and different phase trials for, again, other rare and orphan disease groups like this. So I'm on the board at Bridge the Gap and kind of helping out with some of their research efforts.
Looks like we lost.
I have a niece with SYNGAP, which I'm just excited to be on today to see, obviously, your involvement and listen to what all you all are up to. So thanks for the time.
Thank you and welcome. It's kind of interesting to hear about your work in PT and neuroscience and rehab and gait metrics. That seems to fall quite nicely in line with some of Dr. Fine's work and some things that we're interested in. I think Brad knows why I'm chuckling. It's really because we are family here and we all know each other, but I'm not sure. I guess I'll just, do what I planned on doing.
I texted Leslie to see if there's anyone else who.
Okay, great.
Let you know.
Yeah. So I'm a child neurologist by training, and I direct, and this really is for you, Nathan, because everybody else on this panel they are familiar with me, but I direct the SYNGAP Center of Excellence here at Kennedy, and I also direct the Rett syndrome program. I'm also the medical director for the Center for Autism. I trained in neurology and in addition to that, genetics, and did some neuroscience training. And my interest in research in neurology, neuroscience came about when I was doing my PhD neurology training. And I became really fascinated by the ability of the brain to remap post-hemispherectomy. So effectively, when you take out a hemisphere or part of the hemisphere of the affected side, the remaining hemisphere can remap to then control the same side of the body. And that interest led me to do a postdoc with Paul Worley looking at synaptic plasticity, and then my interest in learning and memory evolved as a consequence. It looks like we have Sereni joining us. Hi, Sereni. I hope I'm pronouncing your your first name appropriately.
Yeah, you're right.
Okay, great. I'm Connie Smith-Hicks. I was just giving a little update on who I am and my path. I'm a child neurologist, and I've been doing work in the SYNGAP space for probably about maybe 3 years now. But just a brief recap, I was saying that I've done my background training was in genetics and neuroscience, and my interest in neuroscience evolved out of my experience in child neurology. Now, while I was doing my postdoc with Paul Worley, one of the challenges that I faced in transitioning from mentored research to independent research was really the fact that my research was in a basic science department, but my academic home was in a clinical department. And the challenge I faced was trying to figure out how to bridge the gap between my basic neuroscience interest and my clinical interest. And that became even more difficult for me when my R01 wasn't funded. And so I became more involved in clinical work, and I began to care for patients with autism. And with time, as you all know, next generation sequencing and precision medicine essentially erupted. And one of my mentors, who is actually also one of Amina's mentor, Ali Fatimi, brought me into the fold of neurogenetics. And that was a tremendous opportunity for me because it allowed me to merge my skills, training, and interests quite nicely. And so I was able to get back into research by collaborating with colleagues at other institutes. I then began to work on projects that allowed me to expand the phenotype of other monogenic disorders. And currently, I've been involved in some clinical trials in the Rett syndrome world. I've been doing some translational research, and I continue to see patients. My research is funded by institutional grants, so some small grants, some foundation grants through pharma, as well as some collaborative NIH projects. So what have I learned throughout this process? As I was thinking about giving this talk, I remembered my postdoc advisor, Paul Worley. I was complaining to him about something or other, and I don't really remember what it was, but I remember him saying, every experience is valuable because there's something you can learn from it. And those words were really very impactful for me, and I think they've certainly helped me over the years. Another lesson that I've learned is the importance of having a skill or tool. And by that, I mean something that you can use to interrogate whatever system that you're working with. It allows you to move seamlessly between your clinic clinical interest and your research interest. I think some of the common ones are EEG, imaging, data science, motion capture, more along the lines of what Amina has been doing. But in addition to that, I've felt that or realized the importance of having mentors. And a lot of times we may feel like, the whole mentorship is overemphasized, but I think it's critical to any career developmental plan, regardless of what space you're working in. And I use the word mentors intentionally because there's never one person who's going to be, everything to anyone. And when I think of mentors, I'm thinking, of content mentors. So folks who understand the field that you're in and can guide you and introduce you to others who are in a similar field, or process mentors. So those individuals who can help support you in obtaining resources or support from your institution or other institutions. And certainly peer mentors, those are also very important. They're often at or above your level. They provide moral support. You can, share ideas of things that work and things that don't. You want to talk about, well, how did you navigate and how much time did you get off from maternity leave and all those things. That's good conversation to have with your peer mentors. I think for mentors, it's one of the questions that folks often ask is, how do I select the mentor from the personal perspective? And mentor-mentee relationship is a dance, but I think it requires that there is honest communication between the two. Excuse me. And I think it's also important that the mentee can be transparent in their communication with mentors. So, I think it's important, for example, that a mentee can say, hey, I'm not sure what the next step should be. This is what I think, what are your thoughts? And can do that without feeling, that their mentor will think less of them. The last topic I want to talk about is the issue of work-life balance. And I believe that's a challenge to many. And I know that it's an area that I am still working on. I have 2 kiddos. One's in high school, the other is in college. And their needs have changed over time. And so has my ability to meet their needs. But one of the things that I've sort of figured out along the way, that it's really important to know when to say no and how to prioritize requests that are made from your department chair. It's also very important to learn how to negotiate. And, I'm still learning that, right? But the good thing is that when you have a large mentorship team made up of a number of different individuals, they can be a great resource to helping inform your decision making. So that's what I have for now, and I'll turn it over to either Jimmy or Brad.
Jimmy, you want me to take it?
Sure, I'll go ahead and take it. Because after that, I'm just going to pepper Brad with questions because I want to get some information from him. So, I'm Jimmy Holder. I think nearly everyone here knows who I am. I'm a child neurologist at Texas Children's Hospital, faculty there in Baylor College of Medicine. Just a little bit about my history, because maybe that's kind of new to people. So I did my undergraduate education at Johns Hopkins, actually, funny enough. And then I went to UT Southwestern for medical school and graduate school. After that, I had a brief period where I was not going to have any more clinical training, I thought. So I went to UCSF for a postdoc. Quickly realized the error of my ways and decided to pursue a child neurology fellowship at Texas Children's Hospital and Baylor College of Medicine. At the end of that, so at the end of that 5-year program, the last 9 months or so, I spent in Huda Zogby's lab, and then continued in her lab for another 3 years or so before obtaining my independence, probably about 4 years actually. So I've been interested for a long time in genetics of brain development and of neurologic disease. I kind of fell into child neurology. I would be curious to hear Brad and Connie's story about how they have found it, but I thought I was going to be either an adult neurologist or adult psychiatrist. I didn't know kids had neurologic problems probably when I, at an early time point in my med school training. But anyway, fell in love with pediatrics and then found child neurology, thankfully, and he took me into the fold and haven't looked back since. So my kind of research career began with, I guess going back to the earliest, my graduate school work was genetics of obesity. And that was, I don't work on that at all anymore. I kind of every once in a while will read a paper about it just out of curiosity. But I had a great mentor, going back to importance of mentorship, who was also a physician scientist, and taught me a lot of things, from how to do rigorous bench research to different types of careers you can have as a physician scientist. And then I would say most recently, in Dr. Zogby's lab, I worked on, I continue to work on a gene called SHANK3, which is also important for synapse development, and mutations in that gene cause a pretty devastating neurodevelopmental disorder, with a lot of similarities in many ways with SYNGAP1, kids with SYNGAP1 mutations. And I'll just, I'm not going to rehash everything Connie said because I agree with all of it. But I would say I totally agree with having multiple mentors. Totally agree with the idea that, and I've heard this from many people, that you really should have mentors throughout your career. So I assume Brad still has mentors that he can call up and, say, hey, whatever, whatever you do at that sort of level. I don't know what you do at that level that he's at, but, really throughout your career. And, to me, it's not just mentorship, it's also having a champion, having that person that really is invested in success of your career. And I think that's so incredibly important. And, I'm fortunate that I can say I have two real champions. My grad school mentors, we're still in contact, and he still asks how I'm doing and what he can do to help. And then Dr. Zogby is continuously, she's really the mother of the NRI here at Baylor, and, really wants to make sure everybody's successful. So I've been very fortunate in that regard. And then, I can't say anything about work-life balance. I'm still trying to figure that out, Connie. I need all the help I can get with that. I have two kids as well, a little bit younger. They're 8 and 7, which, is a challenge in and of itself. My youngest has a neurodevelopmental disorder as well. So that's, another bigger challenge for us. Even as a child neurologist. So, I'm amazed, at the families, what they have to learn and go through when they, when it's really hard for me, even, being trained in the medical field in that area. So in any case, that's it. And I will happily turn it over to Brad.
Well, thanks, Jimmy. Brandon, I see that you joined. So more Kennedy Krieger. But this is a chance for us to, I don't think that for those of you from Kennedy, I don't think that we've actually had a discussion about this topic. So I'll take advantage of the small group and the intimacy of this to tell you a bit about my story and my perspective. And just to out of the blocks with Jimmy's question about mentorship, absolutely. There are people in my life really that have been in mentorship roles for over 30 years now, that we have friendships too, but they're definitely, like, for example, the decision to take this job, there were key people that I turned to when they knew me so well for so long that it was possible to have a really deep discussion, no holds barred, right? Just the straight story. And I think that kind of longitudinal relationship with your mentor, it reminds me a lot of the kind of people who go into child neurology and neurodevelopmental disabilities. Like this, it's at your core to be with somebody for the long haul. And I like that merging of sort of values. But I apologize, I'm going to put up some slides. It's because I wanted to, take advantage of some technology here, but can you see them all? All right. So I'll just blow through that. I saw Adam do his disclosure, so I figured I'd pop one up here. But I thought I'd tell you my story, just like the sort of the origins for me. I did my undergraduate work at Brown. Jimmy, you asked when did we learn about child neurology? I also did not know that there were neurologists for children. But as an undergraduate, my undergraduate advisor at Brown was a guy named John Donahue, who early recognized brain-computer interface type of technology. And he was just a great undergraduate mentor. But it was his wife, Karen Kerman. I don't know if you guys know that name, but she's a child neurologist who at the time was a resident in the MGH program. So the way I learned about the profession of child neurology was from my undergraduate mentor's wife. And the research I was doing in Donahue's lab was presented at the Ether Dome when I was a senior in college. And that's how I knew that I wanted to be a scientist at that point. That was just a dramatic moment. I went to WashU for the MD PhD program. And my graduate work was in basic developmental neuroscience. I studied basic mechanisms in cortical areal differentiation. And then did child neurology residency in St. Louis. And I defended my PhD right before the molecular era hit the telencephalon. So in the early 1990s, there were maybe one or two even transcription factors that had expression patterns in the telencephalon. We were more in the phenomenology, experimental embryology kind of world. And then, so I had to make a decision. Was I going to reinvent myself as a molecular neurobiologist to study cortical development, or was I going to do something different? And I decided to do the latter, which was to dive into neuroimaging. In the late 1990s, there really wasn't much of a developmental cognitive neuroscience to speak of. Only a handful of people had started putting kids in the scanner. It wasn't even clear how to do that kind of work. So the big decision was to retool. I did the K to R type of pathway, K12, K02, R01s going forward. And at the time, the K12 was much more like an individual K award, and the K02 for NINDS was used as a sort of a jump starter, as Adam described it. Other institutes use it differently. Then I ran my laboratory until I came to Kennedy Krieger. But some other decisions I made or I was asked to do in different ways on behalf of the department or the school. One example was that I was asked to direct the residency program. That was a big decision to run the child neurology residency. But back then, it was tenable to do that and be a scientist. But during the period of time that I was running the residency, the ACGME became increasingly hands-on. And so eventually it became clear that physician scientists really can't. They were sort of boxed out from running residency programs, which I think is unfortunate, because in programs that are really directed towards developing physician scientists, I think it's important to have that perspective. And then I ran the NINDS K-12 program for about a decade. The ENSADA programs, that was the original version of the K-12 from the early 90s to 2016. They were institution-based, and WashU had one of them. I trained on it, and then I directed it. And then when I came to Kennedy Krieger, it was around the same time that the K-12 went national, now called the CNCVP, and I am the program director for it now. Co-directed an IDDRC at WashU for 6 years, and then just in the past few months took on the Kennedy Krieger IDDRC. So center director, program director, these are important activities that enhance the work of others. They're not necessarily your own career development, right? It's work that you do to help others in their career advancement. And then eventually said yes to running the division of child neurology at WashU and neurologist in chief at St. Louis Children's Hospital. But for all of you, that I've been saying no to this kind of a job for years. I knew that I didn't want to be the chief, because I wanted to get the research program fully established. But there was also a real push to do other kinds of leadership to serve the division, the department, and the institution. And then the decision to come to this outrageous opportunity to be a Kennedy Krieger a couple of years ago now. So I want to say a few things about what I've learned along the way. One is the point, you can do it all, just not necessarily all at the same time. We'll talk about work-life balance, we'll talk about professional expectations evolving, and then a little bit about mentorship. So this is a really important concept for me from a mentorship standpoint. This gets to the importance of saying no, not yet. Not no, period, no, not yet. The ambitious, smart, ambitious, driven people will want to do the research, lots of clinical care, advocacy, editing this, signing up for that. And my point about this is that one should consider their career as having a trajectory to it, and that eventually you'll be able to do all those things. But if you're going to be a physician scientist, it's very important to dedicate your time to that protected time that you have, and to take full advantage of it to establish your research, and then move into those other areas. I'm not saying that I did it exactly the right way either, but it's an important principle to recognize that there's an entire pathway, and to not be so distracted by the need to do everything all at once. Work-life balance. So I was looking back through old slides. I gave my first work-life balance talk about 15 years ago. And I'm not claiming to have expertise in this area either. But what I like to point out is that I think that the construct may not be the right one. This is what we're typically presented with. There's work and there's life, and they kind of are separate magisteria. And often this kind of diagram then has some arrows. You got to balance the work against life and life against work. So when I see those arrows, I don't think of them as arrows. I think of them as daggers. Like work is attacking life and life is attacking work, that they are sort of at war with each other. Like that it's work versus life. And my view of this construct is that if we start here, you're already in a disadvantageous mode, because when you're working, you're jealous of your life outside of work. And if you're out doing stuff outside of work, you're wondering, when am I going to get back to my productivity? This kind of tension, I think, adds to the negative energy around this idea. So my reframing of this is to think of life as the sphere, all-encompassing, and within that sphere includes family, employment, friends and friendship, other interests, a life of service, and ellipses, to point out other things that you might want to have in your sphere. And the reason why I like this framing is that where does work fit there? Well, for many of us, employment, what we do for a living, is joyful. It's deeply interesting. It feeds the soul because we're pursuing our interests. We think that we're doing work that's going to help other people. So while that might be how you make a living, there's also a great deal of self-fulfillment that can come from it. So employment is not work by itself. My point is that work shows up in all of these domains. Family has work. Friendship can have work. You're pursuing your interests, same. What it takes to really provide, if you're living a service-oriented life, that requires work as well. So I want to turn this idea on its head and reframe it so that we don't have work and life antagonistic with each other. What we have is a comprehensive notion of what living life means, and that all of these elements plus more comprise the experience of life. There's another element, which is time. The dynamics change. Life before the children, life with young children, life with an ill child. I think many of us have experienced all of those chapters, and the dynamics of all those elements that comprise life change over time. And then one more point about work life. If you have a partner or a spouse, it's really important that that person shares this construct. So it's like I dig this construct of life, but if my wife didn't, what would be the point? So thankfully we're on the same page on this. So we used to give work-life balance talks together. So I think it has to do with what your construct is, but also the congruence of that construct with the important people that are along for the ride with you. Professional expectations evolve. I'll make a couple of quick points about this. When I was applying to MD PhD programs in the mid-'80s, if I said, I want to solve Alzheimer's disease, I want to figure out the mechanisms that will make it possible to cure Alzheimer's disease, I would not have been able to get into an MD PhD program. The mindset was, for a physician scientist, even though you were talking about bridging the gap between basic and clinical work, is do good basic work. Don't worry about linking it to your clinical interests. Good things will happen. That's clearly changed. And I think it's important that it's changed. But there was certainly a time, probably going back into the late 90s, early 2000s, but like NIH came up with its blueprint for neuroscience in 2000, that this notion of the pure physician scientist who focuses on basic mechanisms so that good things can happen by others, that's gone to the wayside. The other big one is entrepreneurism. The idea, so we just heard talks from NIH folks about SBIRs, set aside dollars for SBIRs. That's exciting. 20 plus years ago, there was no notion that if you were an academic physician scientist that you should be entertaining the idea of starting a business. It just wasn't part of the expectation and culture. It'd be actually kind of odd to talk about it, a little bit renegade. But about 20 to 25 years ago, that started to shift. Places like WashU and Hopkins a little bit later than other institutions, but clearly the world has changed. Now, I don't know what the next evolution is going to be, but my point here is something different is going to emerge. I don't think anybody in the mid-'80s to mid-'90s would have expected there to be translational neuroscience as a PhD for a neuroscientist, to have the same standing as basic, and that there would be a motivation to start a business, or that was something of value to the academic institution. And then finally, just comments about mentorship. I agree with Connie's points, and Jimmy's points, but I would say that mentorship is also a type of interpersonal relationship. And like any interpersonal relationship, there's not a monolithic way to navigate that space. It really is a dynamic between the mentor and the mentee that has to do with professional development, as has been discussed, with personal development, the former being writing grants and being a better scientist and learning how to collaborate. But the personal development being, how do you navigate difficult terrain, interpersonal terrain in the laboratory or with competitive, people that are competing with you for the same research area. Some of that is professional development, but it's also how do you equip yourself as a person in those settings. That professional and personal development can only really occur in the nurturing environment of a strong interpersonal dynamic that also requires the mentee to accept menteeship, that there's a role that the mentee has to play in making that dynamic work. So Connie talked about transparent discussions. I completely agree. There has to be that real two-way flow of information, a negotiation of what the relationship is like, a negotiation of expectations. And when it works well, decades later, those people are still in your life. So I'll end there and look forward to any questions and further conversation. Thanks.
I guess I have a question, if you don't mind me asking.
Please, Connor.
How do you adapt to sort of new technologies and new sciences? Like you said, genetics came to be sort of relatively recently. And Dr. Smith-Hicks, you touched on how your initial medical training wasn't in genetics. And now genetics has to do with, so many different things. How do you keep up with that while also maintaining, your current responsibilities? Is that just going to conferences every once in a while, reading papers sort of in your free time?
So I guess I'll take that. So I probably forgot to share this bit of information, but my PhD was in molecular genetics back when knockout mice was a thing, and you could get a cell paper just by knocking out, a gene, right? But from a clinical perspective, genetics back then was more linkage analysis. And things have changed now because there's, other than Fragile X and Down syndrome and all these large chromosomal disorders, now we have Rett and Angelman's and SYNGAP and Shank that have point mutations that disrupt gene functioning. And so that has been the biggest change in the field of genetics, right? And with whole exome sequencing, we now have been able to identify the genetic causes for many of these neurodevelopmental disorders that clinically look the same. So in answer to your question, how did I get back in that field? I sort of build on my pre-existing knowledge and read. There are several really great conferences. There is the human genetics meetings that one can go to. And so there are a number of organizations and societies that you can become a part of. You go to those meetings, you read, you just kind of follow the science along and continue to follow your patients, and you see how their phenotype emerges over time.
Great, thank you.
I think it takes a lot of energy and sustained energy. You read, You go to conferences that are outside of your area of expertise, because enlightenment often happens when you hear people thinking deeply about other complicated spaces. And you don't go into these you got to avoid going to these rabbit holes where the entire world, the entire scientific world revolves around your particular topic. I think the way to stay at the crest of the wave is to keep a really open mind and to not be so enamored of your own specific aims page and publications that you think that's the center of the scientific universe. And I know lots of examples. I mean, we could talk about the Fragile X story, where just a couple people sitting next to each other at a conference led to the mGluR5 story opening up because of a couple of smart folks paying attention to each other's talks and seeing an opening. I can't operationalize that. It has to be, I think, just part of your philosophy, or one's philosophy towards science and inquiry.
I have sort of an employment life sphere question. I try to think about it carefully. And I guess a little bit more related to work, but so I've been on faculty basically 3 months before I started leave in October, and sort of already in that short amount of time, there a lot of opportunities came up, exciting opportunities to be involved in basically in every aspect of life that a physician might at Kennedy, between, and I have a primary research project. I have basically like 75% of my time dedicated to research. But, between additional research opportunities, chances to participate in additional like clinics, like multidisciplinary clinics, teaching clinics, basically I think more came up already in that short amount of time than I even expected to hear about. And I personally have a track record of having a hard time saying no to anything exciting. And especially with people that, I admire and enjoy working with. And so any advice for kind of how to navigate what are those things to keep, how to tactfully say no, how to maybe even defer, in a way that the door might remain open. I would appreciate that.
Jimmy, you want to take that one?
Sure, I struggle with the same things. I mean, honestly, I do struggle with some of the same things. I have, I guess there are, I agree with you that, I get very excited about everything in child neurology, right? I mean, I've been at times in my life excited about the idea of specializing in leukodystrophies, at times in muscular dystrophy, I get excited about everything. And, at some point I had to decide, what for me I was going to be most excited about. And then begin to tactfully say, yeah, that's awesome. That's great stuff. I just, I can't do it. I either I can't do it if it's something that, I know that's just not the path that I'm going down for my career, or like you said, say, well, that, or like Brad said, that sounds awesome. Maybe, come back to me in a year, come back to me in a couple of years, and, let me get this established. Because that's, right now I can kind of tell you where I am in my career. Right now, I'm like 99% focused on getting my first R. That's like what I've got to do. And I know that's what I've got to do. And so, I'm pushing that as hard as I can, which means, some other things I'm trying to keep at bay. For example, I've been fortunate to have funding so that I have not had to or have not been on service for 3 years, and I'm trying to keep that at least until I can get that R and get that under my belt and say, okay, now I can begin to incorporate that back into what I'm doing. But these are hard things, right? When you're, for you, when Brad comes and says, "Hey, I need you to," it's hard to say, "Can we," or for me, when it's Gary Clark, right? And he says, "Oh, we really need some coverage," and I've done it sometimes. If it's like, "Hey, we need coverage for a weekend," okay, sure, whatever. But otherwise, I'm like, I really want to focus on getting this R, because that's what's gonna, really set my career on the path that I want to go down. So, that's the best I can give you. Maybe Brad can.
Yeah, so Jimmy, I think all energy towards the R is that's what I would say as well for this stage for you, absolutely. And that's why that bullet point was, you can do it all, just not all at the same time. I think it's a really important message. It's hard, especially if you really like the people that are asking you to do something. It's really hard to not just reinforce the positive moment by saying yes. It's all feels all good at the time. But this phrase "protected time" is, I mean, the way the letters of support from the institution are written, it's, "We will protect Amina's time." But really, having lived through it myself, it's to protect you from yourself. To just do it all. And everybody on this call, there's opportunity. There always will be opportunities. When you say, "I can't do that right now," that's not like, "Oh, that's it. Opportunities are dried up." They will continue to come. That's a promise. I mean, that in child neurology, there's no shortage of leadership positions. There's no shortage of opportunities to help out at the Child Neurology Society. There's no shortage of purely clinical jobs out. I mean, you can configure your time as you see fit. But if you want to be an academic physician scientist, it's, get your independent funding, get your research legs established, then branch out. I think it's really key. And don't worry about the fear of missing out. There'll be plenty of opportunities that come along.
Yeah, I appreciate the advice. And at least, hopefully the pursuit of the independent funding is for a finite period of time, or a grant cycle, and you can kind of, that's reassuring.
So I have a question, and this is maybe, so over the careers, over the career as a physician scientist for a long time, you've seen a large change, I think broadly speaking, because physician training takes so long, you've seen a large change in maybe the clinic, like how clinic is run, the future of clinical care for pediatric neurology. So I think like neuroimaging is probably a great example. And then, so looking to the future of maybe outside of actually clinic, as in you have to come to clinic, but the relationship between patient and clinician outside of the clinic. I mean, we hear this as a basic scientist, we hear about this biometric data, devices that are going to children. And I just wonder, like, when you look at where care will be from my stage, right, before you jump off the diving board, where care will be and how can you think about those interactions for where care will be and how that come back to the science side. I've always kind of thought about that maybe.
Connie, Jimmy, you want to take it? I have thoughts, but I don't want to dominate.
I'll let Connie go.
I was hoping that you would take it, Jimmy. Okay, so to paraphrase, I think how I'm understanding the question is what is the interplay, or how does one use the clinically relevant information that one gets in clinic to inform how we plan for treatment effectiveness or therapeutic interventions. Is that the essence of the question? Did I get it right?
So I think it's that if clinic is going to change as a physician scientist, how can you predict that you should affect your research focus if clinic changes? And are they related, that as clinic changes, so should your research focus, or the questions that you should be asking, or are they completely independent? I think that just more of the trajectory of each, neuroscience and pediatric neurology and how they move forward.
So there is interplay between the two. There generally is historically, and I think in all our work, right? So one informs the next, or ideally that's what we would want to have occur. So for example, if we use SYNGAP as an example, we know that many of the kids have epilepsy, and we know that their phenotype is variable, right? We know that some may benefit from drug X and others may benefit from drug Y. And the question is often, well, why is that the case? And we see that clinically. But a translational researcher or a clinical researcher or a bench scientist may want to explore that to try to identify, are there epigenetic modifiers that make that possible? Is there, other genes in the background that makes that possible? Right. So what we see clinically informs the research questions that we have, and what we identify from the research, we can apply back to the clinic, and so inform maybe the approaches to treatment. Does that answer your question?
Yeah.
So I think I can add a little bit to that. I think I didn't understand the question at first. So I can tell you for child neurology how I was trained and how I think people should be trained from going forward. So how I was trained was really based on phenomenology. So kid comes in with this type of seizure, you give them this type of drug. EEG looks like this pattern, try this type of drug. If that doesn't work, try the next drug. And I'm really, and I think it's starting to change. It's slow. But where we're really driving treatments based on pathophysiology rather than based on phenomenology. And I'm, I get frustrated, now about, teaching people, honestly, maybe Connie and Brad will think differently, but like, teaching Lennox-Gastaut syndrome. It's a compilation of symptoms and EEG markers. And it can be caused by lots of different things. And there's not one treatment, there's not one treatment clearly that works for that compilation of symptoms. And, I think we have to get away from that mindset and get more towards, again, the pathophysiology, which for most of these kids that, we see is going to be genetic. There will be kids with, that had CNS infections at birth or had some sort of trauma or perinatal stroke or something that caused their neurologic dysfunction. But for most of them, it's going to be genetic. And I can just tell you how, the teaching that I do right now is in resident clinic, in their continuity clinic. And it frustrates me that there are still some of my colleagues who, anytime they hear something about genetics, they say, oh, we'll refer to genetics. And so then some of the residents, that's their mindset. If there's a genetic problem, you refer to genetics. And I ask them, well, is it a multi-system disorder that, really needs a comprehensive genetic? No, it's just intellectual disability. Well, you're a neurologist. you should diagnose neurologic disorders. You should figure out what that is. That should be your job. And so you're asking, like, how is the practice evolving? I think that's one way in which it's evolving. Getting away from phenomenology and basing our treatments and diagnostic plans on that to really, saying a kid, this is what I say, a kid has a SYNGAP1 disorder, related brain disorder. Rather than saying, well, this kid has atonic seizures and Doucet syndrome and intellectual disability and listing all these symptoms, rather saying what the pathophysiology is.
Just like to add a little bit to what Jimmy has said. And it's the idea that, many of the epilepsies are channelopathies, right? And so when I think about treating a child with a genetic epilepsy, it's not the clinical phenotype that I see, but what do I know about that channel? And that helps me choose the medications, right? And, one of the things I've often seen is that some kids would be put on sometimes a combination of medications that are all targeting the same pathway. And my strategy is never that, right? It's often to try to target different pathways, different mechanisms of action based also on what we know about the genetics. So I just wanted to throw that in.
It's a great, Brandon, it's a great question, and there's a lot to take on with it. And I think I agree with Connie and Jimmy's takes. I'll say that, so I agree with the framing from the genetic perspective. And Jimmy, frankly, I think what you said, it sounds like we're making a pitch to come over to Kennedy Krieger, but we could talk about that some more at another time. But I think of it as an intersection between the sort of genetic level of mechanism and then as a systems neuroscience problem, because that brain that comes into existence with these genetic issues then has a developmental trajectory that's a cascading interaction between the brain processing the outside world, changing its direction, processing the outside world in that context, and so on. So the ultimate intervention may be informed by a pharmacotherapy or some kind of therapy that's motivated by the proximate genetic issue, but there's this entire systems neuroscience piece that also has to be considered in the intervention. This is why I think giving a 20-year-old a camprosate for their Fragile X is unlikely to just, change their intellectual developmental disability. Maybe in utero, for example. But so that's one idea. And then the other idea to Brendan's question is, I think what's going to happen. So let's take the example of phenotypic variability in thin gap. Where does it come from? I like it. Connie mentioned it. I like this idea that it's in the genetic background. But we don't study the genetic background. We study the gene, and we attribute variability often to a construct of incomplete penetrance to the genetic issue, but it's really likely explained in the background. So our objective is to have the optimized outcome for every patient. If we know that SYNGAP, ultimately what we want to be able to do is read out not just the mutation, but the background, and make powerful predictions about what that individual's pathway is going to look like, and how to take advantage of all that information to individualize their intervention. So I think the future of child neurology, which is both rare and common diseases, and is rare presentations of common diseases, is to learn how to more effectively measure the relevant information that is going to be used to make predictions and interventions on an individualized basis to optimize that patient's outcome. So clinicians are going to have to learn how to, from the scientists, the interaction will emerge as the clinician will have to understand what information is relevant. Trapezius muscle strength on your exam, everybody does it, every exam, probably, or maybe, but for most of what we take care of, it is completely irrelevant. What should we measure for this patient that is relevant, and what can we discard as not really relevant to the patient that's in the room in front of you? I think that's where we're heading. I think that's an exciting prospect for us. So Nathan, I think, had to leave, but one of the points he makes in this comment is saying yes to everything can be painful, but can open doors and make you better discern what you want to say no to. That is definitely the upside of saying yes. And And I'd say, for me personally, looking back over decades, I agree with that. It's just that there were periods of time where, I mean, this maybe is part of personal growth and it worked out well for me, but I know plenty of faculty, being a division chief, I saw plenty of faculty kind of lose their way when it became too much. Or let's suppose they didn't have 75% protected time for research, they had 50%. The likelihood of coming out of an assistant professorship with 50% protected time and getting your R01, it's quite unlikely for the vast majority. So I think making as good a decision as possible, using your mentorship relationships to guide decision making, and to realize that there will be plenty of opportunities in front of you. Saying no doesn't mean saying no, period. It means being as judicious as possible about what really should be part of your portfolio right now versus down the road.
So I have a question for Brad and Connie. So, you know, at least at Baylor, and I think this is an issue beyond Baylor, I think this is an issue in a lot of places. It seems to me like for physician scientists, or I guess let me put it for physician researchers, there's a pretty clear path if you want to go through a basic science, basic translational science pathway where you find a lab at the end of your residency, beginning of your faculty career. It seems like that pathway is much harder to really define for the clinical researcher, the person that wants to do clinical research. And I bring this up for two reasons. One is when I present, you know, what I'm going to present tomorrow, it's basically all clinical research. And the only way I was able to do that was I basically use some of the time that I have dedicated to research in general from K Awards and other grants, and just sneak in the clinical research. And then the other part is I've had some great residents working with me who were, you know, terrific scientists or researchers really, and really should be, you know, now that they've transitioned to faculty, should be doing clinical research, but those jobs are hard to find. They're all doing 100%, 100+% clinical work, and just now cannot squeeze in any research. Do you think that that's a systemic problem, and then how do we get around it?
So Jimmy, we talk about this a lot in the CNCDP. So there are fewer applicants that are kind of K23 oriented than K08. And I think one of the reasons why is that you're laying out that it's harder to do. Often the pathways are not as well blazed at institutions, how to do it. The kind of science that you can do in the bench, the expectations for rigor and reproducibility in a controlled setting is much, there's a deeper understanding of how to do that compared to clinical investigation where you're dealing with you know, rampant variability and sort of a fool's errand of thinking that you can control for all of that rampant variability, but there's some expectation that you'll give it your best shot anyway. I think also there's a bit of a pejorative associated with clinical investigation. I'll say I served on the scientific advisory board. I won't share which one, where it was clear that around the table, this vanguard of neuroscientists definitely viewed patient-oriented or human research, human investigation as a level down from bench investigation, just frankly. So you ask if it's systemic, I think it is a systemic issue. So what do you do about it? So a lot of places came up with these masters of science and clinical investigation so you can understand how to do human research, not just clinical trial, just human investigation per se. But I think there has to be a recognition at study section, at program, at the NIH, at institutions that it's harder. It takes more time to build up your skill set. And you just have no control. You have limited control over the variability that's going to come your way with clinically oriented investigation. Connie, what's your experience been?
My experience has been that it's really dependent on the institution and the departments. I think Kennedy does a terrific job of clinical investigator and supporting trainees in that regard. I think that, as I think back to my own experience, I think my challenge was certainly being in a basic science lab and being in a clinical department. And, the two didn't, the folks spoke together, but the language, the ability to translate ideas between one and the next was not there. And so the clinicians in a clinical department have an understanding of how to do clinical research. We think of stroke related research, right? And the bench scientists have an understanding of how to do that. So I think a lot really depends on where you are, and if you're in that place that where there is leadership that allows for the adequate translation between the two.
Thanks, everyone. I'll have to hop off now. I'll try to come back before the end if I can. Take care. See you soon.
You're welcome. Thanks, Amina.
Kennedy Krieger is definitely discrepant from other places that way. The principles of phenotyping, commitment to deep phenotyping, a commitment to full characterization of outcomes. It's got a very different feel from what's available other places. So I think that's it's a real advantage to the sort of long-standing culture that exists here, Jimmy. But I think it's clinical departments do. I completely agree, kind of, that there's cultures within clinical departments where there's they know how to do the work. The concern that comes up is when a basic oriented review panel looks at patient oriented or clinically oriented work. That's where I think the issues arise with second tier, a view of second tier status from a scientific rigor standpoint, unfortunately. And I think that we have work to do to make sure that the scientific community understands that it's a different set of constraints that you're dealing with when you're working with patient populations.
Well, and that's what I worry. I think especially for neurodevelopmental disorders, in contrast to stroke, right, where part of the issue is just the patient size, right? We're dealing, with the bigger population I work with, right, with Phelan-McDermid syndrome, there are 1,000 kids diagnosed in the U.S. as opposed to SYNGAP, where there's 100 kids in the U.S. diagnosed. But in either case, it's next to impossible to run the same to perform the same sort of clinical trials, for example, or even to understand the breadth and depth of the phenotypes. And like you said, it was nice to hear the NIH program officers speak, but I still came away from that feeling like, okay, what Connie and I have talked about round and round is we need a real prospective natural history study for SYNGAP. What is the mechanism for doing that? You know, I look at the NIH and I'm not sure I see it. Maybe I'm missing it, but you know, I'm not 100% sure I see it.
I don't have the answer. I mean, Connie, maybe you can speak to it from like a Rhett perspective. I mean, but I think the natural history study started, and I don't think it's being continued. There aren't a lot of natural history studies for rare diseases. Yes, exactly. I think it's a big, significant lacune in the way that the NIH thinks about the kind of work that has to be done to set the stage for meaningful subsequent work to get at changing the life pathway for individuals with SYNGAP and other rare diseases. Yeah, you know, when you were talking about Lennox-Gastaut, Jimmy, I was thinking one of my favorite things to harp on there, I think that's Lennox-Gastaut is a great one. I just like to bring up Broca and Wernicke aphasia. So for those of you who trained in neurology, like how many of those pure Wernicke aphasias have you ever taken care of, right? But it's in every textbook.
Right.
These, you know, in some cases, mid-19th century constructs are, you know, still drive the framing of how we think about the conditions we treat. Lennox-Gastaut is a good example of that as well. Phenomenology, overgeneralization from a handful of initially described, well-described patients. Yeah. So Brandon, your question, I mean, I think that there's the optimism that we have about rare disease also is that a deep dive into a given rare disease will have generalizable principles that can be applied to others. And I think that's one of the, like, why study a disease that afflicts 1 in 100,000 or 500,000? It's because from it, there's the high likelihood that there will be generalizable principles to apply to other such rare diseases. Do you, Jimmy, Connie, you buy into that?
I do, and I would extend that to what you learn from these rare diseases flows into common diseases. I think studying synapses is incredibly important for Alzheimer's disease.
Yeah.
So, you know, I totally buy that.
And I concur. I mean, even the talks today, you know, about GABA in SYNGAP and GABA in Down syndrome, and, you know, the use of PTZ, we can see that there is a lot of similarities. I mean, one of the things I often tell trainees is that we have, you know, 20,000 genes. Many of them are expressed in the brain. The brain does a handful of things, right? And so all of those genes are funneled into similar pathways. And so once you begin to understand how the various pathways impact the clinical phenotype, then you can begin to see crosstalk or shared mechanisms. So absolutely. Okay. It's 4:15.
Yeah, I think we wondered if we could talk for an hour and a half, and I think we've gone through it, so it's great.
So, so I'm not an expert in SYNGAP, but so what do you guys think is the next significant chapter for SYNGAP? What's on the horizon that most excites SYNGAP investigators right now?
You want to take that, Jimmy?
Do we want to go naive first or last? You can save me the embarrassment if I can do it first, but
Go first.
okay. I think from the perspective of translation, I think the most hype, I'll call it, is for the genetic therapy. That's good. That's definitely been it's definitely the most talked about. I think most resources have been allocated towards that, large picture speaking. But as far as what is next, because it's odd because we kind of know a lot about its biology, which is very different when I look at, you know, all the disorders at Kennedy. It's not the case. So since we know so much about the biology and now genetic therapy, I think that's what's at least presented to us. But I think the next big thing is and I relate this to Dravet, is that our issue is that all the models for Dravet syndrome have tons of seizures, and then they were a really good candidate, I think, for some of the biotech companies to do genetic therapies. But SYNGAP, the models of SYNGAP have different types of seizure burden. So it's that they do have seizures, but they're not this SUDEP phenomenon at a certain age, which they have in the mice. And this is a good model, because if you can extend life, well, with the genetic therapy, well, that's a really strong biomarker. So I think what's interesting is, do we know enough about the patients moving forward that they are a homogeneous group? But now we know there's variability. What's the important part of variability? And I think we talked about it. Dr. Jimmy kind of asked me a question earlier that, can we get just more screening, finding more patients with the disease and having a better history from really early on? I think that would help, at least where I'm trying to use the mouse model to gain insights that can help the clinic. I think just giving pediatric neurologists all of the tools available for them to say, hey, this is the standard of care, or develop a standard of care, or not. And just that we know more about the early life epilepsy and then understanding outcomes later in life all at the same time.
So I think there are several things I could think about that I think are open questions. One of the ones I've been thinking about a lot recently is trying to figure out what is the spectrum for pathogenic mutations in SYNGAP. And, we have a lot of kids now with loss of function mutations, with nonsense and frame shift mutations in the middle of the gene that all have similar phenotypes. I think I've spoken with Connie that she has some kids that have definitely milder phenotypes and probably related to their mutation. And I wonder how far that can be pushed. I wonder, I've heard of individuals, I think we may actually hear from one on Thursday, that are very, very high functioning. And then, it always becomes a question, well, is it really the SYNGAP variant that's driving their phenotype, whatever that is, right? Because when you have a novel variant that you don't really know the impact on the protein, then it becomes unclear if that's really, we put that label on someone, this is a SYNGAP patient, but are they really? And they might be. So I think finding those patients, and then we need to model that somehow and really show that impacts protein function. And then I'm going to be losing the word that I'm going to be. I'm going to tell what I think is important, which is, like Brennan said, there's 20 years of work on what does SYNGAP do. And I don't by any means think we know everything SYNGAP does, but we know a lot, right? One thing that I've been interested recently, and Brad, if you serve on the NIH studies section, hopefully you'll find this interesting in the future, is what regulates SYNGAP, and can we harness those pathways to somehow improve symptoms in patients, whether it's increasing the function of the existing SYNGAP, or what I propose, increasing the abundance by mucking with the normal degradative pathways of SYNGAP. Or, of course, there are the genetic approaches, which some of them, look really elegant and could work well. I don't think gene therapy is going to work for SYNGAP, at least as it's used right now, the vectors that we have now, gene replacement therapy. So those are some of the things that a lot of what I just said is not specific for SYNGAP by any means. Right. So
Jimmy, so the point you made about the novel variant, and you don't know if that particular phenotype, how much is driven by the novel variant. One of the concepts that I've wondered why it hasn't taken hold more significantly in the sort of neurogenetics related fields is, we know IDD, garden variety IDD, can be explained by a rare variant and common variants. Right? We know that there's role to be played. Variance is explained by rare and common variants in different ways. But it's clear from a biological standpoint that you could have a rare variant floating amongst common variants. And that concept does not seem to be in the gestalt of helping to explain why this individual with rare disease, SYNGAP, maybe with a novel variant, but a presumptively pathogenic but not yet previously described variant, why it is that the milieu of common variants in the background that by themselves might be contributing to 2 SDs below the mean for measures of intelligence. Why that framing has not garnered the attention that, I mean, from where I sit, it deserves. So I'm interested in your take on that.
I think it is, I think it's difficult to prove. I mean, I think it's not low hanging fruit when you're writing a grant, right? I mean, it's much easier to say, I'm going to study loss of function in SYNGAP and do these established assays rather than when you start to say, I'm going to study it in the background of other variants. I mean, it's just not low hanging fruit. I think that's why.
Not it in the background. I'm saying the milieu of other common variants in the background, not it as a background mechanism, but
right
in the context. So we know what some of the common variants, we know what those players are. So it's not like you have to be powered to re-demonstrate that those common variants are factors. You can pull them forward as variants of interest in your a priori hypothesis set and say, if they're present and I have a SYNGAP mutation, I'm going to predict a more disrupted development of intelligence, for example.
The challenge is the number of patients that we have at our disposal. And I think it being a rare disorder, we probably just don't have the end to support, looking at the impact of common variants. I think certainly in rare disorders that have larger N's, Rett syndrome and Fragile X are examples. I think that those disorders, that kind of study could be done in those disorders more easily than in SYNGAP.
So I've seen it done in for example, in the work that looks at the female protective effect for intellectual and developmental disabilities, mm-hmm. that you just sort of draw together, you count up rare and common variants per se for all comers. So I'm not sure that it would have to be a specific issue for SYNGAP. It could be going across rare diseases, and just in principle, using that same kind of approach, can you demonstrate the modulatory effect of common variants on the severity of intellectual disability in SYNGAP or other rare diseases? Because my sort of fantasy in this space is that the background helps you understand the organismal resilience to the pathogenic nature of the variant SYNGAP, for example. And it may be that manipulating what SYNGAP would have done had it been normally formed is one way to treat, but another way is to capitalize on the organismal mechanism for resilience that you might find by protective versus risk-producing background. So back to Brendan's question, as we get better at all this, how do you forward all the information that comes from a genomic analysis and understanding the systems? It may be that we, instead of fixing the problem, we enhance the resilience mechanisms as a more of a global approach to protecting the developmental trajectory. It's a little fantastical, but you can imagine how our better use of all the information that's available could turn into interventions that are based upon that richness of information.
I guess.
Go ahead, Con.
So I guess it seems to me, Brad, that you're instead of targeting the gene or the gene function, the idea here is to target the network. whether it's from a pharmacological approach or a pharmacological plus, habilitative therapy approach. And I think, I mean, that's something that I've found very intriguing and something that I think makes a lot of sense when we think about how clinical trials are done, when we just give the drug and we don't supplement that with, habilitative type therapies.
Yep.
I totally agree, and part of the reason for really, I think we should pursue that much more wholeheartedly, is that if there are 5,000 genes that cause autism, intellectual disability, what have you, no matter how good gene therapy ASO approaches are, pharmaceutical companies aren't going to develop, or unlikely, I think, at least right now, to develop treatments for all 5,000. It's because when you have a disorder that affects 1 in 500,000, it becomes not economically feasible for them to really produce that drug. I mean, much less this, put in the R&D to make it, but even just to produce it is not economically feasible anymore. So I agree. I think that's very important.
Absolutely. I think we're gonna have to call it quits. I mean, this has been a great conversation. I've had a lot of fun. Thank you. Yeah, thank you all. I think it's now time for us to join back that main live stream.
All right. See you guys.
All right, see you guys.
Thank you. Have a good one. I guess it's a Zoom link. That's what.
The live